So first, I'm really sad to miss the CTC. It's one of my favorite meetings. I've been going to it since I was a postdoc really early. But thank you, David, Rob, Shaunak, Hal, Megan, Abe, Amelie, Laura, Evan, Gobeni, and Peter for organizing and hosting this thing again in such a great location. So I'm really fortunate to work with a great group of individuals. You heard from a few of them. The work that I'm going to present here is led by Brian Gotham, Glenn Bean, Anshul Chaudhry, Michelle Berger, JC Choi, and Jake Burrell have both contributed and are contributing to it also. And the overall theme of our lab is really to develop better preclinical models. We want to use the mouse much more judiciously, much more in a more translational way to carry out biological research. And the approach that we've taken in the lab is one of computational ethology. The idea is to combine computer vision with machine learning, as you're hearing. The underlying reasons for this is increased reproducibility, replicability through standardized pipelines and objective measures. It allows the reuse of data, that every data that you saw from JC can be analyzed by other people. It's scalable, so I don't know if JC mentioned mentioned specifically, but that data set that you saw is probably close to 100 terabytes, maybe more. It really is a scalable because the machine does the bulk of the hard work. And what we hope to do with these ethological assays that we're not forcing the animal to do a song and a dance, is to have better translatability to human conditions or human tests, especially in the context of, again, preclinical assays. So, we all need models. This is our model. I think we're all kind of in this model, which is we altered genetic shapes, altered nervous system, which then leads to altered behavior. Most labs are really good at studying two of these three layers. So, we're quite good, and probably most people in this audience are quite good at the genetics and the behavior. I'm right now running a machine learning behavior quantification course here, and I tell you 90% of the people are really good here, and they just bypass genetics. But ideally, we'd like to work at all three levels to have kind of meaningful discoveries and impact. A more complicated view of this model is here, where we have genetic circuits that shape neural circuits. These arrows represent pleiotropy. There is no one genetic circuit for one neural circuit, and there is no one neural circuit for a particular type of behavior. Most behaviors that we're interested in, like anxiety, sleep, circadian, social, are latent. We cannot directly study these behaviors. What we can do is measure some output, right? So we measure, for instance, open field time in the corner, or elevated plus mate as a measure of anxiety. There are other labs that will do social behavior, like the three-chamber assay, or wheel running behavior. But really, this is highly arbitrary. A mouse with altered anxiety will have altered social phenotypes, and a mouse that doesn't get along with its buddy, they're going to have sleep circadian changes also. But because the way we kind of do science, we fragment this and we study domains. The promise of ethological approaches and computer vision-based approaches is that we can study everything together in one assay. So JC can look at, sleep, circadian, eating, feeding, social. We need to think about what are the anxiety measures in this ethological assay. So a bulk of the work that we need to do is start to kind of delineate these arrows. And by perturbing genetic circuits and neural circuits, we can then alter behavior. And then those behaviors become kind of valid behaviors for a particular latent state. This then allows us to start to kind of build these kind of derived indices of our specific latent states. And then we can really kind of pound down and do genetics, pharmacology, all these kind of cool stuff. So, we've already heard about our system, which is called JABS. We essentially build this whole pipeline for us. And I'm not going to belabor this point. I just want to make one kind of point here, which is York Winter's company Phenosis is now making versions of this for distribution. So it makes it a lot easier to get the setup. I have no conflicts with this company. We don't get anything back here. And I know at least one lab in Michigan has gotten this, and think Amelie Bod is trying to get this. So that makes kind of access a lot easier. We built for classifier training, JC talked about this, kind of our own active learning system for behavior annotation and classification of classifier building. Anshul Chaudhary in the lab recently finished a thing called the JABS web app. The idea is that you train your behavior classifier, like the feeding or the drinking classifier, you upload it to the web app, and the web app can do a couple of things. One, it allows another lab to just share that classifier, and it allows us to download classifiers that other labs have built. So it's a sharing resource. The second thing Anshul did was he created 3 data sets, JABS 600, 1200, and a BXD data set. And these are 1-hour open field data sets with 600, 1200, and around 1100 animals. And the concept is very similar to Gene Network. The idea is you submit your classifier, the classifier gets inferred on these data sets. You instantly will get, well, not instantly, but at some point down the line, you'll get heritability estimates for that behavior classifier. You'll get genetic correlation with other behavior classifiers that people have deposited, and you'll get a GWAS analysis out of it. So it really, again, trying to work at those 3 levels of genetic, nervous system, and behavior, like, a lot of neuroscientists who are really good at the nervous system and behavior part can start to kind of integrate genetics into their workflow through this type of approach. Yeah, I'll skip this, but these are just some outputs from the web app. So we've used this for not lots, but we've used these for papers, and these are all single animal behavior papers. And we're now kind of switching more gear towards multi-animal. You saw kind of the foundations of it being built. I want to just give one example. I think this is a fun example of the kind of discoveries that can be made when you start actually watching the animals in a holistic way using long-term monitoring. And this is work from Brian and also from Michelle Berger. So the Simons Foundation funded us to essentially create a data set of long-term monitoring for 4 autism mouse models for the study of autism. And this is the FMR1 knockout, the Shank3 knockout, this 16p11 duplication strain, and then this catnap2. And the idea, again, is we have 4 groups of 3 mice, and we just watch them continuously for 4 days. And we test them at 3 and 10 weeks. So this is a large data set, about 150 terabytes of data are there. And then what we proposed to do, well, what we did was essentially, we tracked it. Well, the goal here is to track the data set with our pipeline. And I think, Brian showed you data on the left. I want to kind of point to the data on the right, which is, half the time mice are kind of huddled like this. And this is more of the challenging thing that we have to actually deal with. And once we kind of have this tracking working, which we do now, we can start to then classify behavior. So we're building and have built classifiers for these kind of social behaviors. JC showed you eating, drinking. We'd like to get sleeping, nesting type behaviors also built. I want to show you something. So one of the things and these are things we propose to do in terms of behavior in the Simons data set. And we also propose to look at play behavior because, social autism, that kind of makes sense. It was kind of a risky proposal because play behavior is not something that's has not been observed historically in mice quite a bit. It's really one where it's been seen in rats. Obviously, humans and most mammalian mammals do some sort of play, but in mice so far it hasn't really been observed. Like I would say, high levels of play, because most of the assays in mice are, again, these short-term assays done during the day. So Michelle Berger in the lab really looked deeply into play behavior and kind of classified all the different types of play that's been shown, like social play, locomotor play, exploratory escape play, object-oriented play. And then she started just manually looking by eye in the data set in the BTBR B6 data set. So, and I wanted to show you, she ended up finding was like lots of instances of play. So this is an instance of rough and tumble play. And if you want, I can definitely play these again. This is a chase, but you'll see it's, it's a pretty, this is not aggressive chase because these are all juveniles. This is really kind of, a play kind of chase behavior. And she watched these quite a bit. Jerk behavior is a classic kind of play behavior. You'll see that here. I'll play that one more time because that's really fast behavior. And then she actually also started to observe object play. So here, these mice look like they're fighting with each other, but you'll see a piece of food drops out from one. I don't know if you saw that. There's like a piece of food right there that one mouse picks up. So they're actually fighting over something. So then Michelle is like, oh, okay, that's interesting. So, she had some dogs. So she made a bunch of toys, obviously a dog person here. And then started giving these to mice at different ratios to see if they would start playing with these objects. And I'm just going to show you one video. She has lots of these videos. So what I hope you can see is that they're actually playing with the ball. And you'll see that more because one mouse takes it and starts running away with it. So then she did the hard job here. So what she did was she took a video and it has 3 animals in it. Sorry. Okay. So there are 3 videos here. And then there are 3 animals in each video. So video 1, video 2, video 3. This is not animal 1, 2, 3. It's animal 1, animal 2, animal 3. And then she looked at the first 24 hours and the last 24 hours of this 4-day assay, and she tried to classify all these behaviors just by eye. And it's hard for you to see, but I just want you to kind of pay attention to all these kind of ethograms, these little bouts of play behavior. Play behavior was quite robust in these when she quantitated all these. So these are the individual behaviors. We're seeing representation of pretty much the whole repertoire of behaviors that she had in that original table. So I'll stop there in the sense like our next step here is really start to build classifiers for these and characterize these behaviors in the BTBR strain and that on those autism strains that we've collected. We've collected them, as I said, in adolescent and adults. If this is actually play behavior, we should lose them in the adults because adolescents play and then adults play much less. That's actually the whole kind of idea behind play. So I'll stop there with this. I want to spend a few minutes on the JAX Digital Initiative because we've been building all these tools. JAX has funded a lot of this work internally for us. And JAX has finally kind of invested in a really big way in a digital initiative. And just to remind everyone, JAX is a nonprofit. There is a business side that sells mice, but we are a nonprofit. Just like, University of Tennessee has a hospital and most medical schools are there associated with universities. The money kind of generated from mouse sales and any revenue go into research. I am purely academic, but I am involved with this kind of initiative. And the goal here is to build a digital platform to empower the biomedical community. This is a machine learning and AI-enabled platform for rodent behavior. And the goal is to kind of collect video data 24/7 in home cage environments and to associate with that all the fancy algorithms that you just saw that we've been working on. So just like our JAB system, there's kind of a pipeline here of study design that's cloud-based, data acquisition that's local, happens on hardware that we built. Then all that data gets uploaded to the cloud, and there are these kind of foundational metrics, machine learning-based metrics that are inferred on this data. And then we have really nice kind of data visualization and data access portals. And then finally, we need sort of machine learning platform, right? Because not everyone, like once you do the pose estimation or the segmentation or the bounding boxes, you need to actually do something with that. So there is a really nice kind of a toolbox that's been built, and it's in the cloud. So all this GPU, CPUs are sitting there on the cloud, and you can just access your videos and your data and your algorithms and kind of really operate at scale. So it's kind of a data scientist and machine learning experts best case scenario. Okay, so the digital cages look like this. There's a housing unit. This has integrated lighting, cameras. It's got a HEPA filter in the back. The cage itself is a disposable Innovive rat cage. Okay, so not a mouse cage, a rat cage. These are not real mice, but they're relatively the size of real mice. The idea is we want to use this for both mouse and rat work. The top-down camera, this is a view of that. So the idea is to minimize occlusion. So the water is here, the food is here. There is a wheel running, we call this furniture. We can remove it, put something else in. But the idea is there's a lot of space here for the mouse to do things. At night, the videos look like this. Hopefully you can see this is coming across, but you can really nicely see the mouse's, ears, ear tags, eyes, what they're doing. And this will actually get much better because we actually have more sensitive cameras. The camera technology keeps getting better. But yeah, so this is a nighttime view of the camera of the cage when the mice are active. Let's see if this, okay, so there's a side camera also that provides a much higher resolution of your mouse when the mouse is in view. This idea here is like, if you want to do sort of health checks and things like that, you can start to do that with this side camera. This is scalable. So, this is one of 4 racks that's at Jack's. Each one of these can fit one of those cages that I showed you. After the data is collected and they're kind of in the cloud, there's a foundational ML model. The ML model does bounding boxes on the mice. It does segmentation of the mouse, and it does 5 key points right now. And this is, of course, something that can be extended. But this kind of model, it's a HydroNet model, which is a fancy way of just saying it has these kind of convolutional layers, and then it has this multi-heads that do different tasks. It's a very fast model in the sense we really optimize this for speed because we've got so much data we need to operate on. And the idea then is you use metrics coming out of here to analyze complex behaviors. But all the data that gets collected gets inferred with this HydroNet. We've been running this for about a little over a year. As of March 2023, we had trillions of frames of data at 4K resolution. We've got multiple sites that are collecting data. And the nice thing is it doesn't matter where you are. Like, you could do this at University of Tennessee and Jack's, and all the data ends up in the same location. And then these models get applied, and then there's kind of a uniform web interface to do things downstream. The visualization for the data looks like this. So if you pay attention, this is a web interface here. These up here, this is over a 30-day record. So every one of these ups and downs is one 24-hour period. This box right here is what's being shown in this window. And so the image of the mouse from the top is here, the side is here. And this is looking at, average movement in the cage, wheel occupancy, water dispenser occupancy, food dispenser occupancy. So the idea here is, if you remember like the UCSC browser type, you have these ribbons, essentially, you can just add your data track. So imagine once you have a machine learning model, you just add that as another track, and you can kind of customize this to what you want. Of course, we're social creatures, not just the mice are social. So by pressing this button, you can share the study with anyone, and then they have access to all your data. If you see something really interesting, you can just put a little note here and at a timestamp, it gets marked, to tell your, colleague to look at this or pay attention to something. So there are these kind of social features for us built into this system. And then finally, for us, what the most exciting part is this data science workbench. This is a scalable compute platform. So again, like you have these trillions of frames of data across multiple sites sitting there, and this then allows us to start to operate on that. It's very much like notebook driven, but the nice thing is it just kind of integrates with the data that that's being produced in the JAX digital ecosystem. So the way we kind of imagine, there'll be more information on this in Q4. There is a website already. The way we kind of envision this is multiple, groups of people that are going to be interested in this. So right now, the biggest group is really the developers. These are the engineers, the hardware, the software engineers that are really developing the system so we can actually get interesting metrics out of this, the foundational models and things like that. The next group, I think, are going to be the researchers, and that's like labs like us, like you. And the idea here is, we want to start to replace those assays. So now we want to do interesting new behavior assays and validate those. And then finally, once we have kind of these new behavior assays and they're validated by the researchers, then I imagine there would be like a large group of users, and these would be like pharma companies, other labs that may not be kind of developers or really, behaviorists, but they just want to do their assay, they want to do their work. And so we imagine like, this kind of building this ecosystem for kind of multiple interests happening. We want to keep this as an open platform for people to really innovate, collaborate on. And I'm really kind of excited about this whole initiative. It's been kind of in stealth mode, and finally, like the results are there and it's working really well. Finally, I'll stop, but I want to have one slide, which is the title of my talk, Homes and Hotels, because we kind of presented two solutions. I just talked to you about the digital initiative, which is more kind of a home cage monitoring system. And then you heard really good work from Gautam and Brian and JC around hotels, which, these kind of open field arenas that you get a lot more information on. And I see these as complementary systems. Mice can be monitored long-term in homes, but maybe at lower resolution. Maybe you don't get gait, maybe you don't get all things you do in kind of the larger arena, but then you move them out into the larger arena on a periodic basis to really, get that high resolution data that's needed. So we should really think about this as a complementary set of systems to accomplish the same task, which is, again, for me, kind of the genetics, the nervous system, and the behavior, and then to improve and have better preclinical models. With that, I'll stop.
Thank you very much.